Hello everyone, my name is Flannan and my student ID is 2025197345. Today I'm going to present the AGR499 course, and I'm going to present the subtopic in the chapter 2, which is the 2.1 Light and Dark Reactions of Photosynthesis, on how plants power the planet. Today, we're diving into one of the most fundamental biological processes on Earth, which is photosynthesis. Specifically, we will explore how plants convert sunlight into chemical energy through the light-dependent and light-independent, which is the Calvin cycle reactions. By the end of this presentation, you will understand not just how, but also why this process is critical for life as we know it. Photosynthesis is a process in which green plants, algae, and certain bacteria convert light energy into chemical energy. It takes place in chloroplasts and results in the production of glucose and oxygen from carbon dioxide and water. Why should you care? How does this biological alchemy transform photons into food, fuel, and the air we breathe? Picture this. Right now, across Earth's green mantle, trillions of chloroplasts are performing quantum physics, splitting water with sunlight, crafting sugars from thin air. This isn't just botany, it's the ultimate energy conversion system, refined over 3 billion years. Today, we'll dissect its two core engines, the light-dependent reaction that captures solar energy and the Calvin cycle that builds life from carbon dioxide. Now we move on to the photosynthesis equation. At its core, photosynthesis is a redox reaction. It combines carbon dioxide and water to produce glucose and oxygen, using sunlight as the energy source. But this equation is deceptively simple. It glosses over the intricate machinery inside chloroplasts that makes this possible. Think of it like summarizing an entire factory with just the input and output. Over the next few minutes, we'll open the factory doors and see how the magic happens. Now, let's move on to the chloroplast structure. Here's where the action happens. The chloroplasts. Notice these stacked disc-like structures? These are thylakoids, the solar panels of the plant. They house the light-dependent reactions. The surrounding fluid, the stoma, is where the Calvin cycle occurs. This partial separation is crucial. It allows the plant to efficiently manage energy capture and sugar production, much like an assembly line with specialized station. For the light reactions, let's zoom into the thylakoid membrane. When a photon strikes photosystem 2, the P680, it excites an electron, kicking off a chain reaction. This electron travels down an electron transport chain, losing energy in small steps, like a ball rolling downstairs. The energy pumps protons H+ into the thylakoid space, creating a gradient. Meanwhile, to replace the lost electron, PS2 splits water, releasing oxygen as a byproduct, the very oxygen we breathe. The electron eventually reaches the photosystem 1, which is the P700, where another photon gives it a second boost. The energy is used to produce NADPH, a key electron carrier, and that proton gradient, it drives ATP synthase, a molecular turbine that generates ATP. Together, ATP and NADPH are the energy currency for the next phase, which is the Calvin cycle. Next, we move on to the cyclic versus non-cyclic electron flow. So, plants are master energy economists. Normally, they use non-cyclic flow to produce both ATP and NADPH, but sometimes, like during stress or high energy demand, they switch to cyclic flow. Here, electrons loop back to the cytochrome complex, pumping extra protons to make more ATP without NADPH. It's like a factory rerouting resources to meet urgent deadlines. This flexibility is key to plant survival in changing environments. Now, let's move on to the stroma for the Kelvin cycle. Phase 1. Carbon fixation, Rubisco, the most abundant enzyme on Earth, attaches carbon dioxide to RUBP. But here's the catch, Rubisco is slow and error-prone. It sometimes fixes oxygen instead of carbon dioxide, leading to photorespiration, a costly mistake. Yet, without Rubisco, life as we know it wouldn't exist. In the phase 2, ATP and NADPH from the light reactions convert 3-PGA into G3P, a sugar precursor. Phase 3 recycles RUBP to keep the cycles running. For every 3 carbon dioxide molecules fixed, only one 3G3P exits the cycle. The rest regenerate RUBP. It's a meticulous process, but it's how plants build the sugars that fuel ecosystems. Why does this matter? The tiny G3P molecule is the foundation of life. It becomes the bread that we eat, the cotton in our clothes, and even the biofuel powering our cars. Photosynthesis also scrubs carbon dioxide from the atmosphere. Without it, Earth's carbon dioxide levels would be 10 times higher. And here's a sobering thought. Rubisco inefficiency limits crop yields. Scientists are now engineering super Rubisco to boost food production for potential game changer for the global hunger. In closing, photosynthesis is more than a plant phenomenon. It's the foundation of Earth's habitability. From its ancient origins to modern lab-engineered improvements, this process exemplifies nature's ingenuity. Yet its limitations reveal opportunities. Could we create a Rubisco that never photorespires? Design crops that photosynthesize at night? These are not sci-fi fantasies. They are active research areas where your generation will live. As we continue to unravel photosynthesis secret, remember, we're not just studying plants, but we're also learning to sustain life itself. That's all from me. Thank you for watching my presentation.